Hi, everybody. I am really excited to be here today. I am Yingji, and I work in CLD cell line development here at Samsung Biologics CDO development. Today, I'd like to walk through our CLD capabilities to enhance both quality and productivity. Today's webinar is structured into three main sections. First, we will start with an overview of current industry trends and the challenges. Then we will dive into our approaches in CLD and USP. And finally, we will wrap up with a brief conclusion. Before we get into the technical details, I'd like to briefly highlight a few industry trends that are shaping how we approach CLD and USP today. First, biologics are getting more complex. With more biospecific and complex molecules in development, we are seeing increased demand for precision in both gene integration and process control. Second, timelines are getting tighter. Companies are under pressure to move from DNA to IND faster, and that's where scalable CLD and intensified USP like N-1 perfusion come into play. And finally, regulatory expectations continues to rise. There is a stronger focus on securities like glycosylation, titer, and potency. So early control strategies are becoming essential. With these trends in mind, let's take a look at how Samsung Biologics is addressing them through our integrated platform. As shown on the left, our CLD has several platforms. We offer the Asteroids platform for transient expression, built upon our proprietary Asteroids cell line. In addition, we have developed the Asteroids SA future host cell line, which enhances ADCC activity. In the overall CLD process, development is progressing using the transposase system. If you look at the track record on the right, about 44% of our projects are monoclonal antibody, while around 56% involve complex molecules. Typical development timelines are about 2 to 4 months for the molecular antibodies and 3 to 4 months for complex molecules. As mentioned earlier, we have significantly improved both productivity and quality by incorporating a transposase system. This system offers several advantages, including single copy integration at each target site, multiple insertion points across the genome, stable integration of the gene of interest, maintenance of structural integrity, and representative bulk pools. When we combined our proprietary Asteroids cell line with the transposase system, we saw a significant boost in productivity. As you can see here, titers increased 5 to 10 times at the pull stage and 3 to 7 times at the clone stage. This productivity gain was not only seen in monoclonal antibodies, but also for bispecific antibodies. On the left, you can see that the transposase system results in higher productivity quality compared to conventional random integration systems. On the right, we see a dramatic increase in the likelihood of selecting high-performing clones. Looking at one of the most important factors in CLD process, which is stability, it can be confirmed that when cells are maintained up to 60 PDR in the transposase system, titer, QP, and quality are also maintained. In other words, it can demonstrate that the excellent cell line stability. Looking at our historical stability data, over 90–70% of our cell lines demonstrate long-term stability. Of those, about 83% maintain over 19% of their original productivity. So in summary, our transposase-based platform not only enhances productivity and quality, but also ensures robust stability early in development. With that, I will hand it over to Sojeong to introduce how we take things further upstream with our intensified USP platform, Intensify.
Thanks, Yingji. Hi, I'm Sojeong from Upstream at Samsung Biologics. So now that we've covered CLD, let's take a look at how we enhance productivity even further using our intensified upstream platform, Extensify. Traditional fat batch processes are widely used in the biopharmaceutical industry for producing a range of biologics due to their simplicity and well-established protocols. In the fat batch process, cells are cultured in a bioreactor, and nutrients are periodically added to sustain cell growth and production. This method is favored for its ease of use and compatibility with existing infrastructure, making it an accessible choice for many manufacturers. However, as we scale up using traditional fat batch approaches, several limitations become apparent. The accumulation of metabolic waste and depletion of critical nutrients in the bioreactor can create toxic conditions that stress the cells, reducing their viability and productivity. On top of that, these suboptimal conditions can negatively impact product quality, which is critical in biopharmaceutical applications. Also, the reactive nature of traditional fat batch processes means we are often adjusting nutrient levels and managing waste after problems arise, rather than preventing them beforehand. This results in inconsistent production and quality variability, which becomes a challenge at large scale. To address this, the industry is increasingly turning to innovative approaches such as perfusion and intensified fat batch. These methods offer greater control over the cell culture environment through continuous or semi-continuous nutrient feeding waste removal. This not only supports higher cell densities, but also creates a more stable and productive environment. The intensified fat batch approach in particular combines the advantage of both traditional fat batch and perfusion systems. It results in higher productivity and better control over the culture environment without the added costs of full perfusion systems. So again, as we scale up with traditional fat batch, some challenges become clear. The accumulation of metabolic waste and depletion of key nutrients in the bioreactor can lead to toxic conditions that stress the cells, ultimately reducing their viability and productivity. These less than the ideal conditions can also compromise product quality, which is critical for biopharma applications. Furthermore, the static nature of fat batch processes means that adjustments to nutrient levels and waste management are often reactive rather than proactive. This can cause variability in production runs and inconsistency in product quality, which poses significant challenges in large-scale manufacturing. That's where the intensified fat batch approach offers a valuable solution. It combines the advantages of traditional fat batch processes with some benefits of perfusion systems, providing higher productivity and better control over the culture environment, all without the high costs of full perfusion systems. Recognizing the need for more efficient and scalable solutions, Samsung Biologics has developed Extensify, a proprietary platform that leverages the intensified fat batch strategy. Extensify is specifically designed to overcome the limitations of traditional fat batch methods by optimizing cell culture density for high-quality production. What makes Extensify unique is the integration of an N-1 perfusion step. This unique step allows us to achieve significantly higher cell densities in the N-1 stage, which means we start the main production culture with a much higher initial cell density. This supports faster exponential growth and enhanced productivity from the very beginning. So with Extensify, we're able to boost productivity while maintaining the product quality. Now, productivity is just one side of the story. To talk about how we optimize the therapeutic functionality, I will hand it over back to Yingji.
Thanks, Sojeong. As she mentioned, while it is critical, therapeutic functionality also plays an important role. Glycosylation has become a major critical quality attribute for many therapeutics currently on the market or in development. Post-translational modifications in proteins, including glycosylation, play essential roles throughout the protein life cycle, affecting folding, stability, activity, and distribution. Controlling glycosylation early in development is key to improving properties like pharmacokinetics, pharmacodynamics, and reducing immunogenicity. To address this, Samsung Biologics developed the future cell line, which produces fucose-free antibodies with enhanced ADCC activity. This cell line not only maintains high productivity and stability, but also enables functional optimization to better meet client-specific needs. As you can see in the left figure, SA Future was developed through S-choice-based cell line engineering. We knocked out the FUTA gene to enable expression of fucose-free antibodies. When we compared the performance of SA Future to the original S-choice cell line, we saw similar growth and productivity profiles. And importantly, A Future was maintained excellent stability just like the S-choice. Looking at the fucose levels in molecules expression from SA Future, we confirmed a dramatic reduction in fucosylation compared to S-choice, effectively in achieving a true fucose-free level. As seen in the right panel, when we evaluated ADCC activity, we saw a significant increase in the antibodies produced by SA Future. Since glycosylation is a critical quality factor, selecting high-performing cell lines based on glycan profiles early in development is ideal. However, early-stage glycosylation analysis is challenging due to factors like small sample volumes, low concentrations, and the purification limitations. To address this, Samsung Biologics developed a high-throughput assay that enables early screening of relative sialic acid levels in HGCF samples. This method offers a high specificity, simple protocols, compatibility with low concentration samples, and a quick turnaround. As you can see in the right figure, the relative binding rate drops dramatically when neuraminidase is added, indicating strong specificity for sialylated molecules. In the left figure, we found a strong correlation between high-performance analytical methods and our lectin results, with positive trends observed across different molecules. For two different molecules, higher lectin binding rates aligned with higher levels of sialylation. In the right figure for case 3 and 4, we observed that sialic acid levels ranked high in both the poor and the clone stages. This means the relative levels observed early in pools are maintained later in clones. Based on this data, we demonstrated that our novel high-throughput lectin assay is effective for screening large numbers of pools and clones with high specificity. By applying this assay early in clone selection, we can screen for optimal glycosylation profiles from the very beginning. Now to show how we take this even further in Upstream, I will pass it back to Sojeong.
Thanks, Yingji. As Yingji said, glycosylation plays a crucial role both in the safety and efficacy of biopharmaceuticals and is primarily determined by the host cell characteristics and the cell culture conditions. With accelerated development timelines, optimizing glycosylation profiles within these constraints is becoming increasingly challenging. S-Clean is designed to address this challenge by enhancing the quality of the molecule, including improving ADCC, CDC functions, as well as productivity. S-Clean achieved this through several steps, including the media and additive screening, process parameter optimization, and analysis of required quality, including ADCC and CDC assays. Finally, the design of experiments, DOE, is applied to optimize the glycan profiles. This platform has demonstrated its efficiency in not only obtaining the most optimal glycans that meet the client-specific needs, but also enhancing ADCC and CDC functions as a result. Since S-Glean is integrated into the standard upstream service timeline, it doesn't require extra time, which enables more in-depth quality analysis and reduced development risks. So we would like to share two different case studies. We used two distinct cell lines that produce two different antibodies using the EMBER-15 system. Firstly, case study A shows the optimization of N-glycans in a fusion-derived monoclonal antibody. Media and chemical additive candidates were screened and chosen that showed no negative impact on relevant cell culture aspects. Then process parameters were optimized using DOE and were further purified and analyzed for N-glycan profiling. Individual N-glycan peaks were identified prior to N-glycan analysis in advance. All data, including cell culture profiles, titer, purity, and N-glycan results, were further analyzed statistically to find the optimal parameters for both titer and N-glycan. This statistical analysis revealed the relationship between parameters and the N-glycan profile, hence providing a clear understanding for determining the best outcome. Finally, we then assessed whether the optimized N-glycans has functional impact specifically on ADC and CDC. Two levels of relevant glycan samples were analyzed to identify the correlation between glycan and effector functions, which was demonstrated to be positive in case study A. For case study A, evidently both ADCC and CDC have been elevated by increasing the levels of afucosylation and galactosylation respectively. In the corresponding graphs B and C in both ADCC and CDC, while the maximum bioluminescence response was increased, the EC50, the concentration required to achieve half the maximum response, has been decreased, which indicates less of the molecule is required to elicit a certain response. Case study B focused specifically on optimizing fucose levels in a monoclonal antibody. By increasing the afucosylation level, we successfully enhanced ADCC activity, again confirming the direct impact of glycan control on therapeutic function. In conclusion, we, Samsung Biologics CDO, are committed to staying proactive and advancing forward by developing cutting-edge technologies continuously. In this webinar, we introduced our enhanced cell line development strategy using transposase for accelerated stable cell line generation. Upstream's extensified technology could further improve the productivity of the stable cell line generated by our CLD group. In terms of product quality modulation, we launched SA-FuCho, which showed enhanced ADCC effector function. S-Glean and S-OptiCharge can be leveraged to control the product quality of the molecule through accumulated know-how and advanced process optimization.
All right, thank you so much. Great presentation. I do have a few questions for you. So first is, how does the timeline change when using a transposase system compared to a random integration method?
When using the transposase system, the simple molecule takes 2 months and the complex molecule takes 3 months. This is a decrease of about 30% compared to the random system.
All right, thanks, Yingji. Another one here. Does the lectin assay only monitor silation levels, or can it also detect other types of glycosylation?
Based on the lectin assay, not only silation, but also galactosylation, monosylation, and fucosylation can be monitored.
All right, thank you. Is scaling up possible if Extensify is incorporated within Samsung Biologics?
Of course, our company has extensive experience in seamless tech transfer from CDO scale to GMP scales. So we also have hands-on experience of N-perfusion in GMP scale, which means we have secured capability to go through late phase, such as phase two or 3. So based on Samsung Biologics technological expertise and experience, the intensified fat batch platform process developed by us is expected to be successfully applied to the GMP scale.